In this video, I'll be covering what is known as the adapter hypothesis, proposed by Francis Crick. Before I talk about this hypothesis, it's important to know about the central dogma, the process by which DNA is converted into functional molecules that allow for essential bodily functions to occur. Here we can see how the information carried by DNA nucleic acids are transferred to RNA nucleic acids, and RNA molecules utilize tRNA to transfer its information to produce proteins. The adapter hypothesis was introduced in order to try to explain how this transfer of information from nucleic acids is used to specify amino acid sequences in an organism. I'll be using the example of a charging cord, a charging block, and its respective socket to represent this hypothesis. Here, the end of this USB-C cord can represent an amino acid, which is the product of translation, and the outlet in the wall can represent an mRNA codon. We can see that structurally the outlet and the USB-C cord are not compatible. This is parallel to the mRNA codons and their respective amino acids, which differ as depicted. We can see that the amino acids have characteristic side chains that vary from being hydrophobic to hydrophilic, whereas nucleic acids are characterized by hydrogen bonding between nitrogenous bases. This means that nucleic acids are good at recognizing other nucleic acids, which is why information can be passed from DNA to RNA, but lack the ability to recognize amino acids. This is where Crick's proposal comes in. mRNA molecules are able to pass their information to code for amino acids through an adapter molecule, represented by the charging cube shown here, which would be able to connect the USB-C cord, which represents the amino acid, to the socket, which represents the mRNA codon. It was later discovered that tRNA acts as the conduit of transfer for information. tRNA is a nucleic acid that contains a 3-nucleotide anticodon region on one end of the molecule and an amino acid acceptor on the other side. An enzyme known as amino acyl synthetase links tRNA molecules with their respective amino acids in a process known as tRNA charging. In order for translation to occur, the tRNA molecules bind to the small subunit of the ribosome, which is a complex of proteins and rRNA molecules. This complex binds to the 5' end of mRNA. Next, the anticodon region of the initiator tRNA binds to the start codon region of the mRNA. Finally, the large ribosomal subunit is added, with each codon of the mRNA, a new tRNA carrying the appropriate amino acid is brought to the ribosomal complex, and the polypeptide chain continues to be elongated. To account for the fact that there are many more codons than amino acids, the wobble effect allows a single tRNA molecule carrying a single amino acid to code for more than one codon. There are many different ways that tRNA would not be able to act as an adapter molecule. Let's say, for example, a mutation in the DNA strand occurs, changing the coding for threonine, ACC, to a stop codon, UGA. Because tRNA is unable to recognize a stop codon, translation would end, which would result in a shortened and dysfunctional polypeptide product, which would likely not be useful for the cell and could cause a multitude of cellular issues.